Welcome back. In this lecture, we'll discuss the algae biofuel industry, my research in algae biofuel, and we'll look at the first step in creating a model, which is inputting the data. So the algae biofuel industry is still surviving. The cost of production is still higher than fossil fuels and other competing biofuels such as soybean oil. But there is an increased need for biofuel. So if they can get the algae biofuel production within the right cost range, then they'll have a market. The University of Arizona conducted DOE research projects for algae biofuel for 8 years. And my role was designing and building bioreactors and modeling algae growth and algae raceway temperature. And there are several important parameters in green algae growth coefficients, light, temperature, salinity, pH, nutrients. But in this short class exercise, we'll look at simplified versions of two parameters that feed into the growth coefficient, light and nutrients. So production of algae for biofuel was first popularized at UC Berkeley in the 1970s. And in the late 2000s, with the concern over global warming, interest in biofuels became high. And with high government and private funding, there were dozens of algae biofuel companies in 2010 with millions, tens of millions, or hundreds of millions in investment. But they have almost all gone out of business. The reason is that the technical readiness level was about 5, and people were basing their business plans on selling algae in the marketplace. The technical readiness level of algae is currently between 6 and 7. Nevertheless, with the market for biofuel in the US, there is hope, and there are a few companies that are continuing to conduct research in algae biofuel. And I think it's sort of like solar energy, which was not economical for decades, but has recently become a better economic alternative. The Department of Energy invested research money in solar for many decades as scientists worked to bring it up to the TRL 8. And a similar thing is happening with algae. Scientists and engineers make small advances, and it eventually might compete with fossil fuels or even other sources of biofuel. So this is one of the algae companies that's left. Synthetic Genomics was started by Craig Ventnor, who was involved in the Human Genome Project. He's pretty famous. And it was funded by Exxon beginning, I think, in 2004. And their focus was on genetic engineering of high-yield algae. Recently, the name was changed to Viridose, and they have large algae raceways like this one in the California desert. And in this picture, the paddle wheel on the left pushes the algae around the raceway. It's shaped like a car raceway, which is why they call it a raceway. And the reason that they keep the algae moving is they're mixing the algae and keeping algae moving up into the light and so on. Exxon recently pulled out of funding Viridose, but Viridose raised $25 million last year to continue their research and production. So there's a lot of interest in this technology. United Airlines was one of the investors. They invested $5 million because they have a high interest in being able to fly their jets without causing global warming. And other companies are also committing to an algae or a biofuel future anyway. So Viridose has a very productive algae strain. I heard that they produce 36 grams per meter squared per day. So that would be for every meter squared of raceway, they produce 36 grams of algae at the ASU, Arizona State University test beds in midsummer. And I think they're in the range of 18 grams per meter squared per day for the entire year, which is really amazing. And don't quote me on that, but I think that's what I heard. We were down, if you look at the whole year and the crashes due to predators and so on, I think we were down in the 5 grams per meter squared per day production rate in our little raceways. Reliance Energy in India is the largest energy company in the world and also the largest algae biofuel company in the world. and they're very aggressively pursuing algae research and production. And I remember that they participated in the Department of Energy algae biofuel programs that we were involved in, and they adopted some of the technologies. The big push at Veridos and Reliance is to grow algae on seawater and to use land that is unsuitable for farming. And Reliance has a big algae production facility on the Indian Ocean. The reason India is an attractive place to grow algae is that the cost of manual labor is extremely low. And there is a lot of manual labor involved in algae production. Two other algae companies that are still in business are Global Algae Innovations and Microbioengineering. Global Algae Innovations is led by a real visionary who has about 8 acres in Hawaii. And then Microbioengineering is led by pretty much the leader of, I'm not sure he's still the leader, but he was the leader of algae research for many years. And also our own Dr. Coelho has an active algae research program. He designs these inexpensive bioreactors, and he has focused more on algae for food or pharmaceuticals or other bioproducts. But I think he's now growing algae for biofuel. This is the global algae farm in Hawaii. And instead of a paddle wheel, they have this two-part sort of half raceway, and they'll lift the water up here and allow it to flow down, and it just flows down to this point, and then they have a pump that lifts the water up. I can't remember if it's an airlift pump or a propeller pump, but that's a pretty nice system. With this system, they're able to keep the depth pretty low, so that even though the algae culture might not allow light to penetrate more than 1 or 2 centimeters, if the depth is low, then you don't have a lot of algae in the dark. These are little raceways produced by Microbioengineering. They're in San Luis Obispo. And so we use these for experiments. So these have been pretty popular over the years, where you just conduct experiments in experimental size raceways, which is much easier to manage, you can imagine, than a giant raceway, if all you're interested in is characterizing growth coefficients and so on. So I looked up a recent paper for costs for algae biofuel production, and it said that the cost per kilogram of biodiesel production for algae biofuel is 0.459 dollars per kilogram, which is $1.5 per gallon. I'm kind of surprised by that. I might have calculated something wrong. But you can see that most of the cost is cultivation, moving the algae around the raceway. And then extraction, that is the process where they convert, or they take the oil out of the algae, and they do it with methanol in this case. And then there's transesterification, where they break down the fatty acids into biodiesel. And then they have, they also produce glycerin by this process, and then they have methanol recovery. There's other methods for extracting algae, fuel from algae, such as hydrothermal liquefaction and several other methods. And so when you do a cost analysis, you have to consider all the costs. So here they're mixing the fertilizer with the water. Here's the algae raceways. Here's a centrifuge where they're removing the algae from the water. But even in a centrifuge, there is still a lot of water left. It's maybe 10% solid. So then they still have to dry it. And then I don't know exactly how they're extracting it. They're using methanol here, which breaks down the cell. And then they do transesterification, where they break down the these fatty acid chains. So they get rid of this backbone here. And then these chains are about the right length for biodiesel. And then there's all sorts of cleanup processes, and so I don't know what they all are. But anyway, in their cost analysis, they included all those. But what you can see here is it's a pretty complex process. And for example, the centrifuge uses a lot of energy. So there's always research going on trying to improve, like getting it out by filtration or settling, or different ways to reduce the energy. And you can imagine you make little minor changes here, 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 here. They add up to maybe 4% improvement. And that's just how this type of research goes. So here's from the same paper, the cost of resources for algae production, and they said that the highest cost was the fertilizer. And then the environmental impacts in millipoints, which is sort of like carbon equivalents, but just a different measurement method. So these are the environmental impacts. You have a cost of producing the fertilizer, and you have environmental impacts of that. So you have to consider all these things in what's called life cycle analysis. Okay, let's start focusing on the algae. So algae are eukaryotes, not like binary fission with prokaryotes. And green algae are classified as chlorophyceae, and plants are similar. In fact, green algae are sometimes included in the plant kingdom, and plants are sometimes included in the algae kingdom. So green algae conduct cell division at night, and they undergo what's called multiple fission. So they have multiple offspring. And in undergoing this process, they lose biomass at night. They're exerting energy and losing biomass. And so this is just a simple diagram, not really, of multiple fission. And what you can see is that there's growth phases, and then there's what's called a commitment point. If the algae is big enough, then it's going to divide, or it's going to go ahead and make that commitment to divide. Then it's going to undergo pre-synthesis and synthesis, where it's producing RNA and proteins, and then it's going to grow some more, and then it's going to undergo mitosis, and so on. And you can see that the algae just keeps dividing, and you might have 8 or even 16, I think, algae produced. If you have three divisions, would be 8 algae daughter cells. And I'll show you why I think maybe there's 4. Anyway, this is one of the algae species that we grew. This is chlorella, so it's a type of green algae. And it's one of the most popular types of algae for biofuel production because it has a high growth rate. This is another algae that we grew. It's called Synodesmus, and you can see that this has 16 cells. So you would imagine that it underwent division 4 times. If it underwent division 3 times, it would only have 8 cells. Synodesmus does not, the daughter cells stay connected by the cell walls, so they don't divide out. This is nice because Synodesmus settles very fast. So if you do want to use settling to harvest your algae, you could use Synodesmus for that. Here's another type of Synodesmus. Okay, and then another thing you have to worry about, which is really a big deal, is predators. 2 of the worst ones are ciliates. So here are ciliates, and they're called protozoa. They're not animals, they're single cells, but they just kind of scoop the algae into holes in their structure and consume algae. And then these are actually animals. They're only about 1,000 cells, but they have these little like vacuum cleaner things on the end, and they just keep scooping algae into their mouths so they can eat a lot of algae. Rotifers. Okay, so like I said, the algae concentration decreases at night. So when you simulate it, you have to have a daytime growth coefficient, and a nighttime growth coefficient or reduction coefficient. So C represents cells. So this is the concentration of the algae. And then you also have to characterize the light. And we're just going to use a simple model. We're just going to use a sine curve to characterize the solar radiation. And then we're going to reduce the solar radiation as the concentration goes up. So that's why this is the effect of solar radiation. And we're also going to keep track of the nutrients, nitrogen, phosphorus, and carbon dioxide. You can see the carbon dioxide is going down, and so it's going to actually lead to death phase. So it reaches this stable phase, and that's just due to light. There's not enough light to grow anywhere beyond this. And so anyway, that's what we'll be modeling. But like I said, we're going to start with data input. And an Excel spreadsheet can be treated like a database if you have fields and records. So fields are the columns and records are the rows. And the way that Pandas data frame reads the data in, it will automatically use the first line as the field names, and then it'll read in the next lines as array. Well, I don't know if you'd call them arrays, but the whole table is keyed to these field names. So what I want you to do is put in the data in your Excel spreadsheet, make your own Excel spreadsheet, put in the data, and give the data these names, and we'll see what these cells represent, or fields represent. And then what you're going to do is you're going to give the spreadsheet the name algae input data. And what happens is every time you change it, we're going to use it in OneDrive, you download it and then upload it again, and then it changes the name to like 1, 2, 3. So you'll have to change the name sometimes. But this is actually very nice because then you can keep track of where you are, which spreadsheet you're using. It just keeps things really organized. So what I like to do is make my spreadsheet in OneDrive, and then I download it from my files to the computer, and then I upload it to Colab. Now, you might not be using Colab for your Python programming, but if you don't, you can just put the Excel file in the same file as your code if it's on your computer. And then you don't need to use this path. You can just give the name of the file. Okay, and so we're going to give it the path where the file is, and then we're going to read it in. And PD, you can see that I imported pandas as PD. So this makes input data a pandas data frame. And this is one of the functions in pandas is read Excel. We give it the Excel path, and we're going to tell it the sheet name. So you need to name your sheet inputs, or else change this to sheet 1, so it knows which sheet to read in. And we're going to give it the sim name as class sim2. That's going to tell the program in the lines below which row to read in. Okay, so this is really nice because you can set up different simulations in your spreadsheet, and then you can just refer to the row that you want to analyze. So go ahead and do that. Turn off the video, and then once you get all that working, once you can see that you can read in your spreadsheet, then turn it back on. Okay, welcome back. And I guess I should have shown this before, but here I have algae input data in my OneDrive, and I have it in my BVE 423 2024 file. You have to be in my files in order to download. You can't do it from home in OneDrive. So anyway, I'm in my files, and now I could just click the download. I click these 3 dots and click the download button, and it'll download it to my computer. And then you go to sample data, and you click the 3 dots over here, and you say upload, and you can upload the file from your computer. You can see that I've uploaded several versions. So this was my first one, this was my second one, this is my third one. And so now, go ahead and, well, hopefully you already did that. But anyway, if you haven't done it already, go ahead and upload it now. And then you can see that I have input data, which is both rows of data. Now I'm going to do input in, where I'm going to select one row of the data. And the way I'm going to do that is I'm going to say input data simulation name. So remember, that was a field, simulation name. I'm going to say input data.loc, and loc means which row we're looking at. input data.loc, it's going to take input data simulation name equals sim name. So here's sim name right here. And anyway, I've said class sim2 there. I could have said class sim if I wanted the first row. Okay, and then you're going to read your data from the row that you picked into variables. And so, for example, here's my time steps. Well, my time step is this long. It's actually, I said equals 1/24 because it's 1/24 of a day. And so I'm going to say dt equals float, input in. Remember, I'm getting my row. And then this is my field name. And I have to say float. If I don't, it reads in a list like 0,0.0416. And so anyway, I struggled with this for a bit, and you often have to. But once you get it working, it's working. But anyway, I found out that I had to say float, 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 making it a floating point number rather than a list. And I also had to say nt was int. So anyway, you can copy what I did here, and then I'd like you to make your own inputs there. So all you're going to do is just put in the name of the fields. Okay, and then the last thing I'd like you to do is put in your initial concentrations of nitrogen, phosphorus, and carbon dioxide. So it's like you added fertilizer at the beginning of the simulation, but we're just going to say that their concentrations are there. And so anyway, you make these vectors or numpy arrays for these concentrations starting at 0, and then we're going to say that n at 0 equals n0. But we didn't read in n0, p0, or CO2 So what I'd like you to do is go back to your spreadsheet and add some columns over here for n0, p0, and CO2 and then make some input lines for those. We'll see you next time.